Hi, everyone. We're going to give a couple more minutes to let everybody connect, and then we'll start the seminar. Okay, I think it's time to start. So hello, everyone. I'm Bruno Cuevas. I'm postdoc at UW Madison, and today is my pleasure to introduce the last seminar of the year for this live RCN. During this year, we have a lot of amazing talks. We have talked about different topics from microbiology to the exploration of icy moons. And today, to conclude this wonderful year, we have the pleasure to introduce our invited lecturer, Maxwell Lechte. So Maxwell Lechte got his higher education in the University of Melbourne in the Department of Geological Science, or Earth Science, sorry. And today he's gonna introduce us to how eukaryotes could have interacted with oxygen at the beginning. So as I say, thanks everyone, and we'll have the seminar and then a small round of questions and answers. So Max, the floor is yours.
Great, thanks so much, Bruno, and thanks so much everyone for coming today and for inviting me to talk. It's a really exciting seminar series to be a part of. I've been following along for a little bit now, and there's been some really great talks so far. So yeah, I'll just share my screen. And get that up. How does that look?
Looks good.
Looks okay. Yeah. Okay, great. So yeah, as mentioned, I'm going to be talking a little bit today about early eukaryotes and their relationship with oxygen and what we can understand about early eukaryotic evolution from looking at the geological record. So this has been a really fun project to be a part of. It's really a team project. So I've listed the other team members here. This has been funded by the Simons-Moore joint collaboration on the origin of the eukaryotic cell. So that's also been a really, there's a broader project. There's a lot of other research groups funded on this same call, mostly biology groups. And so we're kind of bringing the geological perspective to tackling some of these questions about the origin of eukaryotes. And I just want to emphasize, in addition to the other team members, so Maggie Whalen, Galen Halverson, Angela Dos Santos, and Leanne Reedman and Susanna Porter, in particular, this work that I'm going to be talking about today. I'm going to be talking a lot about fossils. I'm actually not a paleontologist. I just want to make that clear from the start, I'm a geologist. And so these little fossils that I'm going to be showing here, this is all the work of Leanne Reedman. And so this work involved looking at literally over 10,000 fossils, and that was all Leanne, so it wouldn't have been possible without her. So I'll kind of make references to that throughout. So getting into it, the evolution of Earth's surface systems and the biosphere have been inextricably linked. So we know that the Earth has transitioned from the early Earth, which had an anoxic ocean and atmosphere system and an entirely prokaryotic biosphere. And then eventually we transferred into a Earth that is more familiar to us today, where we have the well-oxygenated ocean and atmosphere system, and we have a complex eukaryotic biosphere. And there are a few steps along the way. So we know that at some point, cyanobacteria evolved the capability to perform oxygenic photosynthesis, whereby they can split water using sunlight and produce and to fix carbon, as well as producing oxygen. And then we also know that at some point, aerobic respiration evolved, which is bacteria essentially performing the back reaction, where they're using this food, which is being fixed by this essentially limitless source of energy from the sun, and combining it with oxygen, burning that fuel to fuel their metabolism. And then at some point, we also know that prokaryotes must have undergone some kind of symbiogenesis in order to produce the eukaryotic cell, which is this more complicated cell, which has a nucleus as well as a mitochondrion, which fuels the metabolism of eukaryotes. So we know that this evolution of the eukaryotic cell, it completely transformed and terraformed our planet and led to us today. So it was one of the most fundamental evolutionary events in all of Earth history. And so what do we know about this important event? A lot of what we understand about eukaryogenesis is based upon molecular phylogenomics and phylogenetic studies of living eukaryotes. So what we can do is reconstruct the tree of eukaryotes and look at their genetics and their characters that they have in common, and kind of work backwards. And we know that all eukaryotes, including all of us, plants and fungi, and all other animals, all share a common ancestor. And we actually know a fair bit about this last eukaryotic common ancestor, which we kind of affectionately refer to as Lechte. Based on these phylogenetic studies, we know that they must have had all of these quite complex eukaryotic features that are preserved throughout the eukaryotes. So we have things like the nucleus, the complex cytoskeleton, endomembrane system. But the one that I really want to focus on today for this talk is the mitochondria, which again is what permits aerobic respiration in eukaryotes, and also the expression of larger genomes, which allows eukaryotes to evolve into more complex forms. And again, so aerobic respiration is really what's fueling these complex eukaryotes and permits the evolution of large body size. And in terms of an astrobiological spin, it is, of course, goes without saying, it is essential for intelligence and civilization. So humans being the only life form that we know of that has undergone a search for extraterrestrial intelligence, and also is the only life form that we know of that would likely to be have a civilization that's remotely detectable. This is all fueled by oxygen and aerobic respiration and its acquisition by eukaryotes. So yes, anaerobic eukaryotes do exist, and they are widespread and diverse, but they all descended from, again, the same common ancestor that had full oxidative capacity. So because of this, it's kind of long been assumed that oxygen and aerobic respiration played a foundational role in eukaryogenesis. You could say that eukaryotes are addicted to oxygen. And so it's actually, we can go back to the work of Lynn Margulis, at least in the 60s, where it was suggested that perhaps all of these events were actually directly linked, whereby oxygenic photosynthesis produced oxygen, and then this led to the great oxidation event, whereby the atmosphere transitioned from anoxic to oxygenated. And then this is really what drove the acquisition of the mitochondria and therefore eukaryogenesis. However, in the intervening decades, there's been some other studies that have kind of cast some doubt on some of these aspects of this eukaryogenesis model. Geochemists can look at marine sedimentary rocks and their chemistry, and in order to try and work out the chemistry and the oxygen levels of the water column in which they were deposited. And a lot of work on Precambrian marine sedimentary rocks is kind of converging on this model whereby oxygen levels were much lower than what they are now. And they may have even been as low as around 1% of modern atmospheric levels. And this is intriguing because if you get to this level around 1%, it's actually getting around what we refer to as the Pasteur point, which is the point at which facultative aerobes switch from aerobic metabolism to anaerobic metabolism. So it's been speculated that maybe if early eukaryotes evolved under this backdrop of low oxygen levels, perhaps they actually were not respiring aerobically as the main form driving their metabolism during early evolution. But also, if we think of eukaryogenesis as the acquisition of all of these complex eukaryotic characters, we know that they must have been acquired in a certain order, and it's debated whether or not the mitochondrion was acquired first or whether it was acquired after the acquisition of some other complex eukaryotic characters. And so we can kind of envisage these hypothetical so-called stem eukaryotes, which might have had some eukaryotic complexity, but actually lacked mitochondria, in which case they might have been anaerobic. So what was the role of O2 in early eukaryote evolution? That's kind of the overall question that I'm going to be talking about today. So here is a kind of a very schematic tree of life. I know that there's a lot of biologists in the audience. Again, I'm a geologist. Don't focus on this too much. It's really schematic. But what we know that life started with a last universal common ancestor and split up into the two broad prokaryotic groups of the bacteria and the archaea. And biologists argue a lot about the reconstruction of the tree of life, but people generally agree that you can place eukarya within the archaea, probably within a group called the asgards, and that the mitochondria was probably acquired from the bacteria, was likely some form of alpha proteobacteria. And so based on, again, molecular phylogenomic studies, we can try and infer what these ancestors probably were. It's believed that the ancestor of the mitochondrion was probably an alpha proteobacteria that was likely capable of aerobic respiration. And then there's been a lot of really exciting work coming out, especially in recent years, on what the archaeal ancestor of eukaryotes was. We think, again, within the asgards. More recently, it's been suggested that it's within the group called the hods. And some of these, intriguingly, inhabit oxic seawater, and they might involve oxygen within their metabolism at some point. However, a lot of other asgards inhabit anoxic marine sediments and other anoxic environments and have anaerobic and symbiotic relationships within these settings. So it's kind of unclear what the metabolic basis was for eukaryogenesis. So again, going back to this tree, we can kind of think about this in a really schematic way, where we can identify this node, which is Lechte, the last common ancestor of all eukaryotes, and all of its descendants we refer to as the crown group eukaryotes. So this includes some extinct lineages marked here with a little X. But then after, prior to the evolution of Lechte, we might have had hypothetically some organisms of eukaryotic complexity that might have had some, but not all of the features that are characteristic of Leche may or may not have had a mitochondrion, and we refer to these as stem eukaryotes that postdate their first eukaryotic common ancestor. But because by definition all of these stem eukaryotes are extinct, we can't speak to them by looking at modern organisms. We really need to look at the geological record, and that is the fossil record. So again, I'm not a paleontologist, but I have worked with paleontologists, and you can kind of broadly summarize 3 different approaches to Precambrian paleontology. Ideally, you can go out into the field, you can look at outcrops, and you can find body fossils, carbonaceous compressions, and so on. If you can look at macroscopic things. So here's an example of a green algal fossil, which is about 1 billion years old. This is some of Katie Maloney's work, and that's kind of ideal and exciting when you can do that, but they're quite rare in the Precambrian. So more commonly, we rely on petrography, looking down a microscope and looking at chemical sediments such as carbonate or chert rocks. Here's a kind of silicified carbonate, and then if you look at it, if you cut the rock so that it's so thin that light can be transmitted through it, you can look at it under a microscope and you can see things like these filamentous microfossils, which are probably some kind of cyanobacteria. But the approach that Leanne uses and is quite common at the moment for Precambrian micropaleontology is acid maceration of mudstones, mudstones being fine-grained siliciclastic rocks that accumulate in low-energy environments. And if you take those mudstones and you dissolve them in acid, what's left behind is this organic residue, which hopefully can have some nice fossils, as shown here. So I just want to kind of quickly spin through roughly about 4 billion years of the fossil record in a couple of minutes, just for those who are not so familiar with the order of events. So we know that life has been around on Earth since pretty much the beginning. So some of the oldest rocks that we have on Earth have evidence for life in them, which I've always thought is really cool. And that's kind of another big question that I'm not going to touch on today, but these early fossils are things like filamentous forms or simple organic walled microfossils, these kind of spherical forms that don't really have any distinguishing features that we can reliably use to identify what we're really looking at. And so because they're so simple, they might be prokaryotes. This record continues for quite a while, for hundreds of millions of years. And then we get, eventually in the Proterozoic, we start to see some macroscopic fossils. And these are really enigmatic. It's that they're large enough to see with the naked eye, and they're clearly some kind of biological feature, but paleontologists argue a lot about these. I'm not really going to touch on them because we really have no idea what they are. They might be potentially some kind of eukaryote, which would be quite strange because they're really old, but they might also be some kind of colonial organism. So I'm going to kind of brush over those today. And it's not till the near Proterozoic, about 1 billion years ago, that the fossil record, we first start to see things that we can actually recognize and place into modern groups. So the so-called crown group eukaryotes. And about 1 billion years ago, we start to see these really nice algal fossils, for example, these red and green algae. And so this shows that because we have not just eukaryotes, but these red and green algae, we know that this must postdate eukaryogenesis, and that we must have, a record of eukaryotes going back even further. And so that's really what I want to focus on today in terms of the fossil record. Or actually, yeah, I'll just quickly add that for those who are interested in biomarkers, which is the record of fossil organic molecules, we can find these diagnostic steranes, which are considered to be really reliable indicators of eukaryotic primary production, but we don't find those until after the oldest crown group eukaryotes. So while there are some things in the biomarker record that have been recently referred to as protosterols that might be some kind of precursor to eukaryotic sterols, they might also potentially have been produced by prokaryotes. So there's no real kind of smoking gun as of yet in the Proterozoic. So we need to stick to the fossil record. And so I'm going to be talking today about these complex organic walled microfossils. So we have, starting at about 1.7 billion years ago, we have this really rich fossil record of these organic walled microfossils that have sufficient complexity, unlike the older ones, to be able to say confidently that they have no clear analog within the prokaryotes. So we think that they're of eukaryotic complexity because they have things like plates and scales, which is considered to likely indicate the activity of intracellular vesicles. They have complex ornamentation on the cell wall, which probably requires a complex cytoskeleton. And they also have some features which might indicate the capacity to form cysts or pilomes. So some complex reproductive strategies as well. And these, again, start about 1.7, which is right around when you look at the molecular clock estimates for when eukaryogenesis probably occurred. It's kind of right within that window. So we're probably looking at some form of eukaryote. And if you look at this and the backdrop of what oxygen levels were probably like at this time. So in parallel to paleontologists kind of reconstructing this fossil record of eukaryotes, we have geochemists trying to work out and arguing endlessly over what oxygen levels were probably like at this time. So especially during the Proterozoic, as you can see here, this is starting from really low levels, essentially no oxygen in the archaeon, and eventually getting to modern levels. But the Proterozoic is the most uncertain time in Earth's history. So intriguingly, as I said earlier, it might have been as low as the Pasteur point or even lower. So at the lowermost levels, or the estimates for the Proterozoic, this would potentially cause some problems for aerobic organisms during this time. So this is interesting in terms of trying to think about what these organisms represent and what their metabolisms might have been. So while we have these complex organic walled microfossils and people generally agree that they're probably not prokaryotes, it's quite hard to place them on this tree. So are they crown eukaryotes and therefore had mitochondria? Or are they a stem eukaryote that also had a mitochondria and might have been aerobic? Or do they predate the mitochondrial endosymbiosis? Or perhaps, even more controversially, are they just some kind of complex archaea? The more exciting work that's coming out on these Asgards, we're learning more and more about how complex these prokaryotic ancestors probably were. So what can we say about the geological context of these fossils in order to try and understand a bit better about their life modes and metabolisms? So what were the habitats of these oldest eukaryote fossils, and did they inhabit anoxic or oxygenated environments? It's kind of a simple question, and thankfully it's one that we can get at using relatively basic geochemical, sedimentological, and paleontological techniques. So as I said, one of the main ways to look at Precambrian fossils is to do acid maceration of shales or mudstones. And so this produces this organic residue and the microfossils, but also it's actually the exact same rock type that is really suitable for what we refer to generally as shale geochemistry. So looking at the chemistry of these shales can tell you about the chemistry of the overlying water column. And this is a really widely used tool because shales can be found in a wide range of environments. They're found all over the globe. And you can look at things like here, which is ion speciation, which is a commonly used tool, which looks at the ratio of highly reactive ion relative to the total ion in the sample, and also looks at the amount of pyrite in the sample. And this can tell us whether the mudstone was probably deposited under an oxygenated water column, or whether it was an anoxic water column that might have had free iron or free sulfide in the water column. And this is because under oxic conditions, iron is really poorly soluble and becomes more soluble under oxic conditions, but has a really high affinity for sulfide in forming things like pyrite. You can also look at the redox-sensitive trace elements in these shales, and so looking at the kind of redox cycling of these elements on the modern Earth and the biogeochemical cycles, we can try and infer based on the abundance of, or potentially the stable isotopes of these redox-sensitive trace elements, try and understand a bit more about what was going on in terms of redox. So this is really important because you can take the same sample that had the fossil in it and say, we can speak directly to this local redox environment rather than just speculating generally about background atmospheric levels and so on. So because we can do this approach based on the same samples and allocate a fossil to an actual redox environment, at least theoretically, this is a framework that was laid out in Susanna Porter, who's one of the PIs on this project, in a 2020 paper, where you can have these kind of hypothetical predictions for what we might see in the fossil record based upon different inferred metabolisms of early eukaryotic evolution. So if theoretically you had some anaerobic stem eukaryotes, or perhaps they were actually crown eukaryotes, but that early in their evolution, they were facultatively aerobic, but preferred anoxic environments, and eukaryotes were absent from oxic environments, then we would expect to see much more diversity and abundance of eukaryotic fossils in anoxic samples than those in oxic samples. Whereas in a post-mitochondrial world, say if all eukaryotes were obligately aerobic, or they just preferred aerobic environments, then we would expect to see the opposite. We would see much more diversity in oxic samples. Whereas if early eukaryotes were metabolically diverse and they could live anywhere, then we would expect to see a different diversity trend in that case as well. So the important thing about this is that this is testable against the geological record. So the geological record that we're going to test these hypotheses against is the McArthur and Burundadu basins of Northern Australia. So Northern Australia is a really the perfect kind of laboratory to test these hypotheses, because during the Proterozoic, we had the Northern Australia was probably inundated by this large inland sea depicted here on this map, which probably persisted for, in some form, around 500 million years, and was really stable and accumulated all these nice mudstones, trapping lots of nice fossils for us. And importantly, this persisted from about 1.8 to about 1.3 billion years ago. And so again, this is really in that window that's kind of the critical interval that we think that eukaryogenesis might have occurred, or at least early eukaryote evolution might have been going on. And it coincides with the oldest eukaryote microfossils that we know. And thankfully, there are a lot of eukaryotic microfossils within these successions. So collectively, there's really thick sedimentary successions here. There's over 20 kilometers total of stratigraphy if you go from the oldest rocks to the youngest rocks that are preserved there. And they're really well preserved. There's even, they've been essentially unmetamorphosed in most cases, which is really good for the preservation of these fossils, but also for the preservation of geochemical redox signatures. But there's also some base metal mineralization in some parts of the McArthur Basin. And so this means that it's really useful for us because there's been a lot of drill cores drilled through these successions that we can look at, and we can try and find fossils. And also drill cores are really useful because, unlike surface outcrops, which are subject to oxidative weathering, these are probably more likely to have been better preserved in terms of their original geochemical signatures. And as you can see here, I've just plotted up a few of these taxa that are considered to be eukaryotic and their distribution throughout these units. This is our work, or more accurately, Leanne's work in finding these fossils, but this has been shown before. The previous workers have found eukaryotic microfossils throughout these successions. So just a quick summary. I'm not going to really go into too much detail about the fossils and what they indicate, and I'm going to be talking more about their kind of depositional environment and their redox preferences. But just kind of quick summary, it's kind of consistent with previous work, which is shown that you can find these fossils. These are quite common and long-ranging taxa, which are considered to be eukaryotic. So there's things like the plate-bearing Satka favosa, Tapania plana, which has these nice tubular processes, and then also some kind of complex ornamentation and features on the cell walls, such as these longitudinal lineations and concentric striations within Filaria lophostriata. Other things like segmented forms like spiromorpha. So you can kind of classify different eukaryotic fossils, well, I should say different microfossils in terms of how confident paleontologists are that they are eukaryotic. So in order to be conservative for this study, we've listed out form taxa, which we believe are the least controversial in order to reduce the likelihood of incorporating things that might have been prokaryotic and therefore affecting our interpretations. So it should be noted that all of the results that I show from now on are being quite conservative, in that there are probably a lot more eukaryotic fossils of more contentious forms that are not being incorporated into the statistical analyses. But overall, we analyzed a lot of samples. About 3/4 of them were fossiliferous, and about 1/3 of them were eukaryote bearing, which is a pretty good strike rate. Leanne has identified over 12,000 fossils. I have here that the really conservative number of taxa that are being classed as so-called probable eukaryotes is 8. I think I've actually recently changed that to 10, but still working on that. I have to defer to the paleontologists on that for what they can agree on. And there's a lot of specimens of those probable eukaryotic taxa. In addition to those widely found and well-accepted eukaryotic taxa, I just want to give a quick plug to this as a paper led by Leanne that is coming out soon. It's in Press in Paleontology, where she has identified and proposed some new probable eukaryotic taxa, such as the oldest known operculate taxon, which probably indicates the complex reproductive strategy as indicated by this programmed opening of the cell wall. There's wide annulated filamentous forms, which have some cellular differentiation, such as this terminal chamber here, and then also some other platy tubular microfossils, which again, probably require the activity of intracellular vesicles and potentially are evidence of Golgi bodies during this time. So keep an eye out for that paper. So how do we class these fossils in terms of the depositional environment, and how can we reconstruct the habitats that these fossils might have lived in? So ideally, I'm mostly a field geologist, so I like to get out and look at the rocks in outcrop, like this one here, which shows some nice ripples on the surface of what would have been the seafloor at the time. There is outcrop of these basins, and the overall geological context of these interpretations is really building on decades of field-based studies, primarily by researchers at the Northern Territory Geological Service. So if you're interested more in the geological context of these samples, I would refer you to some of these studies and the cited references within. But the drill cores themselves, even though we're not getting the full outcrop, we can still see a lot of really nicely preserved sedimentary features. So for example, you can see these desiccation cracks here, which are really good evidence of subaerial exposure of the surface, similar to what we see in these kind of intertidal zone. And things like cross-lamination, reactivation surfaces, typical tidal features. So we can have reasonably good confidence in our assignment of these mudstones to a specific depositional setting. So we see quite a broad range of depositional environments in the McArthur and Berindadu basins. We range from kind of offshore, probable below storm wave base, where we accumulate these finely laminated organic rich shales, to more shallower deposits, heterolithic facies, right up to coastal environments where we get desiccation cracks indicating periodic subaerial exposure. And we also have the development of these restricted lagoons, which you can accumulate finely laminated sediments embedded with things like stromatolites as shown here. And there's a lot of evidence in some settings for evaporites. So what about the redox conditions of these different environments? Well, so again, the main tools that we used here, the shale geochemistry. So that is both the ion speciation that I mentioned before, which looks at the relative ratio of reactive ion to the total ion in the sample, and also supplementing this with the redox-sensitive trace element analysis. So I'm going to go into that in a bit more detail soon, but this is kind of just a summary of what we see. And the main thing that I want you to take from this is that it's really what we expect to see. This is not really a controversial interpretation in terms of we see these kind of back barrier, really shallow, tidal flat coastal settings are essentially all oxygenated. And that's what we expect because they are so shallow that they would have been, the overlying water column would have been in gas exchange equilibrium, probably with the overlying atmosphere, which we know was oxygenated. The well-mixed shore face zones are also, the vast majority of them are coming up. The geochemistry is showing that they were oxygenated. Whereas as you get more offshore, you get more evidence of the development of anoxia and locally euxinic conditions. What's interesting is that in these kind of deeper restricted settings like subtidal lagoons, we also see the geochemical evidence for the development of anoxic conditions. So this really provides this good laboratory for us to test these different predictions about how this might affect the distribution of eukaryotic fossils. So again, the ion speciation is kind of the main tool that we're using, and then all of this has been supplemented by independent paleoredox proxies, such as the preservation of organic matter and redox-sensitive trace elements, which again all kind of behaves the way we would expect to, where we have these enrichments of these redox-sensitive trace elements under more anoxic conditions. But what's really interesting is that if you look at the distribution of eukaryotic microfossils specifically in these samples, you can see a really strong trend emerges where the eukaryotic microfossils are far more abundant and diverse in samples that are showing up as oxygenated, and those that have geochemical evidence for deposition under an anoxic water column are essentially barren. So this is really striking. But there's a lot of controversy over the interpretation of shale geochemistry. Even though in this case, these are from drill cores, so we can avoid things like debates over surface weathering and metamorphism. There's still a lot of debate about how to interpret, for example, ion speciation signals. But the main point that I want to show here is that even if you throw out all of this geochemistry and you just look at the depositional environment, as I said earlier, these really shallow environments, which is where we're seeing the eukaryotic microfossils being preserved, it would be much more controversial to say that they were anoxic than to say they were oxygenated, because they're just so shallow and they're interacting with that oxygenated atmosphere. So we see a lot of eukaryotic microfossils, a lot more diversity in these shallow coastal, tidal flat, back barrier environments, and also the shallow marine shore face type settings, and really hardly any in the offshore or what we're interpreting as these anoxic lagoons. So if we go back to this kind of hypothetical framework that I brought up earlier about the predictions for the distribution of the eukaryotic microfossil record, the fossil distribution that we're seeing here in the McArthur and Berindadu basins is really more consistent with this model B, rather, the oxic-only model, in that it seems like eukaryotic microfossils are much more abundant and diverse in samples that have a geochemical signal that's showing that they were deposited under an oxic water column. So how do we translate what we're looking at is the abundance and diversity of fossils in a shale. How do we translate that to where those organisms were actually living? Is there some preservational factors at play? What's interesting is that if you instead of just looking at eukaryotes in general, if we look at specific taxa, it appears as though you can see some environmental preferences starting to emerge, which is really a cool result of this study, in that things like Satka actually seems to like the lagoons when they're oxygenated, whereas Tapania you see more in the tidal flat settings and so on. So we think that this kind of supports the inference that the preservation of these fossils is at least to some degree reflecting their environmental distribution at the time that they were living. And what's interesting is that it's not just a general trend going offshore, getting less diverse and less abundant, because in these lagoons, which are, they're subtidal, but they're still relatively near shore, they're really barren in those when they're anoxic. So potentially that's showing that there's something to do with the redox that's controlling the distribution of these fossils. So just a quick recap, eukaryote microfossils are far more abundant and diverse in mudstones that are deposited in nearshore oxygenated environments. A few really important caveats to consider. Have they been transported prior to their burial and preservation? We think it's probably less likely that they've been transported from deeper environments into more coastal environment. It's probably more likely that they would be washed offshore and preserved in the offshore settings if that was the case. But it's definitely something to consider, because when we look at the bulk rock acid macerates, we can't really tell things about transportation very clearly. Is there a preservation bias? Is it to do with oxygen at all, or is it to do with the availability of nutrients or things like salinity? So these are all things to consider. Just a quick comment on preservation. So yes, taphonomic factors are potentially important, but it should be noted that traditionally, at least, we would expect more anoxic settings to limit organic matter degradation, because we're not having your aerobic organic matter oxidation going on in these settings. And when we look at the rare eukaryotic fossils that are preserved in these anoxic samples, they're actually quite well preserved. So broadly, this is consistent with previous studies, which have shown that eukaryotic microfossils tend to increase in diversity and abundance as you go offshore. Although at least in this Butterfield and Chandler study, it was argued that the most proximal, these really coastal tidal flat settings and so on, are actually more dominated by prokaryotes, and that perhaps they were unsuitable for eukaryotes. Although if you look at the analog settings in the modern environments, it's clear that these are thriving and diverse eukaryotic ecosystems from animals, primary producers, and so on. So we believe that they would have been suitable back then as well. So just kind of quickly wrapping up here. Again, it's really interesting. It's getting a bit more arm wavy now, but we can start to think about things like nutrient control, which of course is strongly linked to redox through the redox cycling of different bioessential elements. It was suggested that during the Precambrian, things like phosphorus and nitrogen might have been really limited in offshore environments and might have therefore restricted eukaryotes to nearshore environments. So potentially that is something to consider here. And just to kind of conclude, we can speculate about a lot of things, but what can we actually say about these studies? We can say that eukaryotic microfossils are more abundant and diverse in mudstones that were interpreted to be deposited in an oxygenated water column. And this might suggest that the eukaryotes were at least tolerant of oxygen. Or another possibility to consider is maybe that they were actually living in the sediments and therefore they're decoupled from the water column redox. So that's something to work out as well. And then just finally, just trying to speculate about what we can actually infer from this result in terms of eukaryogenesis and early eukaryote evolution. There was a recent study which suggested that the enzymes for aero tolerance in eukaryotes might have been conveyed by the pre-mitochondrial endosymbiont. And therefore, if that's the case, if we can say that these eukaryotic microfossils were organisms that were aero tolerant, that would suggest that they postdate mitochondrial endosymbiosis, which would be a really cool result. We can speculate about whether the preference for oxygen habitats implies aerobic respiration or not. That's a little bit more tricky, but what we can say for sure is that eukaryogenesis likely began earlier than the Mesoproterozoic, so probably before these oldest fossils that we're seeing. And I'll just leave it there. Thank you. I'll take some questions.
Great, thanks, Maxwell. It's been a great seminar. So I'm gonna go reading some of the questions. So we have a question from Donald Borg, asking approximately how many examples of a given morphotype do you need to observe before you accept that they represent a population of organisms?
Yeah, so how many of each do you need before you can say? Yeah, so it's tricky because historically a lot of this Precambrian paleontology has been done without enough numbers to actually do things like population analysis and statistical analyses and something. So that was actually one of the goals going into this project is, can we try and get enough numbers up to be able to look at things like the distribution? So at the moment, it's really just based on a presence or absence thing. I know that Leanne is really careful in terms of her assignment of microfossils to a specific taxa. So yeah, if she's confident that that taxon is there, then we will say that it was present in that sample. So then we can count the samples, the number of those taxa, and that's probably indicative of something, although then it gets a little bit more uncertain about things like, was it just the right environment in kind of accumulating these things or preserving them? So I would say that presence or absence is probably the main one that we go from, and then it gets a bit more speculative from there.
Okay, great. Second question is, are all of these samples from the same time period, or can you say anything about the timeline? Did the anaerobic eukaryotes appear first, and then was there an explosion in species diversity after the acquisition of mitochondria? So there are a few questions there.
Yeah, so I'll just try and go back to one of the slides which has the timeline on it. So yeah, for the purposes of this talk, I kind of lumped everything in together a bit, although, as I said, we're sampling broadly within this kind of 500 million year interval. So it's a long time. And if I go back to, where was it? This slide. So we have some of our samples are from units that are around 1400 million years old, right back to some that are probably closer to about 1750. So it gets a little, it goes back to the previous question about how many samples do you want and in order to do things like statistical analysis. So yeah, you really want to avoid trying to group together completely different time periods. These are hundreds of million years apart. So, but then the more that you divide it up, the less numbers of fossils you get. So I would say at the moment, we're really probably not quite able to look at things like, the explosion related to, the advent of aerobic respiration and so on. Although I'll redirect you to the paper that I spoke a bit earlier by Leanne, where she's looking at things like, compiling all of the previous studies of Precambrian microfossils and looking at diversity trends and so on throughout the Proterozoic. So these are questions that are being worked on at the moment.
Okay, great. I think we have a new question. Well, I see there is like ongoing discussion also on the chat. There is a more speculative question by Andrew. Well, maybe you could comment a little bit on what you think about this. So considering like he's asking like whether seeing that plants and animals both contain mitochondria and a common ancestry, he's wondering it's like if maybe what is the role of photosynthesis in all this, like whether we should also be thinking about photosynthesis in the to understand the, I guess, the emergence of the mitochondria. Do you have any comment about that?
So I'm just reading the question as well now, just to have a think about it. So that they need photosynthetic oxygen to consume. Did all aerobic eukaryotes have a photosynthetic common ancestor? No. So the photosynthesis was acquired in a subsequent symbiotic event where the plastid was acquired by plants, by algae rather. So they essentially took on a cyanobacteria in order to photosynthesize. So that was a later event that probably took place after the time, or at least the oldest time period that we're looking at here.
Okay, great. Then there is also a question by AC. Is there any speculations about the cell composition of these microfossils?
The cell composition?
Yeah, the shell.
Yeah, so there's some really interesting work that's going on in that space at the moment where these things, they're around sort of 50 microns is probably a typical size for these organic wall microfossils. So they're pretty small, but people, the advances in analytical techniques are leading to opening up a whole lot of possibilities about doing analyses of, taking one specific microfossil and looking at the chemistry of that. So, yeah, I'm not too on top of the literature in that space, but yeah, that's definitely some work that's kind of cutting edge work that's happening at the moment.
Okay. No, okay. I have a question of my own. So in this case, using the geological record, you are able to constrain like the amount of oxygen or amount of sulfide. So I was wondering, could you also, is there any other proxy that you can use to estimate like the primary productivity of those environments? Like how much, for instance, like how much photosynthesis will have been taking place in these tidal environments where you find these eukaryotes?
Yeah, so that kind of speaks a bit to the biomarker record. People have been trying to look at, the proportion of, how much of the primary production is eukaryotic versus prokaryotic in these settings. Unfortunately, we don't have eukaryotic biomarkers from this time period to be able to do that. But just in terms of primary production in general, I guess you could look at things like the organic matter preservation in these samples. So yeah, some of these samples are quite organic rich. Actually, there's actually oil preserved in the McArthur Basin, which is also why a lot of these drill cores have been drilled. So yeah, I guess that would be one way potentially. There's been some more kind of development in proxy space in trying to reconstruct primary productivity, especially in terms of like eukaryotic primary productivity. People have been looking at zinc, for example, some other kind of trace element studies that could be useful. So yeah, we've really started from kind of what we think is the most kind of basic geochemical approaches, just as trying to kind of prove this concept of combining geochemical analyses and paleontological analyses on the same samples and seeing what we can do with that. And it really opens up a whole lot of possibilities of doing more, proxy work on these samples.
Great, seems great and fascinating. Yeah, it's like that's the beginning, right?
Exactly.
Cool. I think we don't have any more questions. So we're close to the time. So thanks, Maxwell, for your excellent talk, and thanks everybody for attending these live RCN seminars this year. Next year we'll come back with more interesting seminars in the intersection of early evolution of biology and all the other fields that we're interested in. And so, yeah, thanks everyone, and I guess Happy New Year.
Yeah, thanks so much for coming and for having me and for all your questions. It was great. I look forward to the next seminar.